Okay, so today I'm going to talk about another recent paper that came out of my lab in 2019. This is a paper looking at the function of ATR kinase. So I've talked about this protein before. It's a protein kinase that regulates various DNA damage responses. Most of what we know about its function is in the context of replicating cells that are moving through the cell cycle. And so in this work, what we did is to look at ATR inhibition specifically in quiescent cells. So these are cells that are not replicating, but can be induced to replicate. And specifically then look at the effects of how inhibiting ATR in quiescent cells affects the response to cisplatin. Cisplatin is a very widely used anti-cancer drug, a very successful compound. And however, and although there's clinical trials being looking at the effect of combining cisplatin with ATR inhibition to improve the effectiveness of cisplatin-based cancer chemotherapies, we don't really know a whole lot about what ATR does and how it may modify cisplatin responses in cells that are not replicating. We think this is important for example, for senescent cell or for quiescent cells that are or stem cells that are not actively replicating, but will nonetheless be damaged by UV or by cisplatin. Sorry. So this work was actually a honors undergraduate thesis project by Rebecca Hutcherson, who was as I said, an undergraduate in the lab at the time. And this was a component of her honors project she did in the biochemistry department. And this paper was published in again, Mutation Research in 2019. So as I mentioned already, the focus of this was to look at what ATR is doing in response to cisplatin treatment in non-replicating cells. And so to initially sort of demonstrate that ATR inhibition will sensitize cells to cisplatin as expected, we took a couple different cell lines, one of which was Hackett keratinocytes. We widely use these in the lab. And then U2S, it's an osteosarcoma cell line, also a very widely used cell line in the field. And so what we did was to pre-treat cells with vehicle DMSO or the ATR inhibitor VE821, a very well characterized, highly specific inhibitor of the ATR kinase. So pre-treat the cells with either vehicle or ATR inhibitor, then expose cells to increasing amounts of cisplatin. And then 2 to 3 days later, or 3 days later here, cells were stained with crystal violet, which is a way of sort of staining cells that remain on the plate and they're likely viable. And so if we look at this, and for example, in the green bar here, this is looking at vehicle-treated cells. Clearly, increasing cisplatin doses causes a decrease in cell survival. However, when cells are treated with the ATR inhibitor, there's even less cell survival. So the cells are sensitized to the drug. We see very similar effects with U2S cells. They seem to be a little bit more resistant to cisplatin than the Hackett cells. Nonetheless, these data show when cells are in a proliferating state that ATR inhibition does indeed sensitize cells to DNA damage induced by cisplatin, consistent with what has been shown in the literature. So we next wanted to use these cells, but in a non-replicating state. So what we typically do in the lab is to essentially grow cells to confluence and then lower the amount of growth factors that they're exposed to. So normally in cell culture, we typically use fetal bovine serum as a source of growth factors to allow cells to proliferate. And typically we use 10%. And so if we lower that down to about 0.5% or sometimes even lower, the cells, especially in a confluent state, that will no longer be able to divide anymore. And so to verify that these cells were indeed in a non-replicating state, or at least a less replicating state, we took either normal subconfluent proliferating cells or cells in a quiescent state grown to confluence and deprived of growth factors, and then pulsed them with BRD. This is a nucleotide analog that gets incorporated into genomic DNA when replication is taking place. And we can detect that by microscopy, or in this case, what we did was immunodot blotting, where we purified the genomic DNA after that labeling, spotted it on a membrane, and using an antibody against BRD to detect that BRD incorporation. And if we look at it, particularly in the Hackett cells in comparison to the proliferating cells, there's very little, essentially no DNA synthesis taking place in these quiescent cells. In U2S cells, it's down still about 5-fold or so. So it does significantly reduce the amount of DNA synthesis taking place in these cells. So we now wanted to simply compare the sensitivity of quiescent and proliferating cells to cisplatin. So both Hackett cells and U2S cells, essentially we took these cells in these two different growth states, and then exposed them to increasing doses of cisplatin. And these results are consistent with what other people have shown in other cell lines over the years. The quiescent cells tend to be quite resistant actually to the cisplatin. So, and this is, I think consistent with what we would expect in terms of why we use drugs like cisplatin for cancer therapy. Cancer cells are typically thought of in terms of a rapidly growing state. and then they become very sensitive to DNA damage, since they're no longer able to replicate their DNA. So if we look at the Hackett cells, for example, this curve shows the cells in the proliferating state, again, very, very sensitive to cisplatin. The cells in the quiescent state are much more resistant. And we see very similar results in the U2S cells. Again, the quiescent cells are much more resistant to cisplatin. So again, the goal of this project is to look at ATR activation and what it's doing in these cells. So we first wanted to validate that ATR does indeed get activated when cells are in a quiescent state. So we took both Hackett cells and U2S cells, pre-treated them with DMSO, again, vehicle, the ATR inhibitor V821, or another ATR inhibitor known as AZD6738. And this compound is actually currently in clinical trials for inhibiting ATR. And so if we look at the vehicle-treated cells, in the absence of presence of cisplatin, and these were treated for 24 hours with either 60 or 30 micromolar cisplatin, depending on the cell line, we can see that using antibody against a phosphorylated form of ATR, we see that phosphorylation level increases with cisplatin treatment. Furthermore, the ATR substrate P53, again, this is a tumor suppressor gene, also becomes phosphorylated after DNA damage induced by cisplatin and other agents. And we can see that phosphorylation increases after cisplatin treatment. However, in cells treated with one of the two different ATR inhibitors now, we don't see that level of phosphorylation being stimulated, therefore suggesting that these inhibitors are indeed inhibiting ATR signaling. That's true both in the Hackett cells and in the U2S cells on the right. In the figure B, essentially all we're doing is quantifying this from several repeat experiments, again, further showing that the ATR inhibitors suppress ATR signaling. So one of the questions in the field is, how is ATR getting activated in non-replicating quiescent cells? So we know a lot about what ATR does in proliferating cells, and the model is that you get, when you have DNA damage, it stalls DNA polymerase movement, but the helicase continues to unwind DNA, therefore leading to long regions of single-stranded DNA that recruit RPA and that recruit ATR to become active. And there's other proteins involved. This is, again, oversimplification. In quiescent cells, again, we don't have any DNA synthesis, or not the same level of chromosomal DNA synthesis for sure. So other data from my lab and others have suggested that a nucleotide excision repair might be important in activating ATR in this state. And so nucleotide excision repair is a system that cuts out bulky DNA adducts, including those induced by cisplatin and by UV, cuts out that DNA in the form, or cuts out the damage in the form of a small DNA. That leaves actually a small gap, about 30 nucleotides in length, in the genome, and that has to be filled in by DNA synthesis. Those gaps have been suggested by others to be a potential signal for activating ATR. And so we wanted to know if ATR signaling that we're observing here in these quiescent cells in response to cisplatin is dependent upon nucleotide excision repair. So we use this drug spironolactone. I have a separate talk about what spironolactone does in a different paper. But it was shown actually a number of years ago to inhibit nucleotide excision repair by causing the degradation of a nucleotide excision repair protein known as XBB. So we can see again in the vehicle-treated cells, we see phosphorylation of ATR and P53. However, cells are treated with spironolactone, that phosphorylation is inhibited. Again, arguing that the ATR signaling taking place in quiescent cells is dependent upon nucleotide excision repair. So what is the function of ATR? What happens if we inhibit ATR in these cells? And one of the more common things to look at when people do these sorts of studies is look at survival. How does it affect cell viability? So we took, again, Hackett and U2S cells, pre-treated with DMSO or this ATR inhibitor V821, then exposed the cells to increasing doses of cisplatin, and then looked at cell survival by crystal violet staining. And so, just as we showed earlier, again, increasing doses of cisplatin, we see less and less survival. However, when cells are treated with the ATR inhibitor, they are sensitized to the cisplatin. So it's actually quite similar to what we observe in replicating cells and proliferating cells. So it looks like ATR has a pro-survival function in response to cisplatin treatment in quiescent cells. Now this is using V821. Just to validate that this is true with other ATR inhibitors, we used the AZD6738 compound, this AstraZeneca drug. We similarly show that inhibiting ATR does sensitize these quiescent cells to cisplatin. Now, cisplatin is one member of a family of platinating agents that are used in the clinic to treat different types of cancer. So we also treated cells with carboplatin, another, as I said, another drug in this family. And we see very similar things. Carboplatin cells are actually it takes higher doses of carboplatin to actually induce cell lethality. But nonetheless, we see that inhibiting ATR, both in the Hackett cells and in the U2S cells, sensitizes the cells to carboplatin. So we observe what we observe with cisplatin, we also see with carboplatin. Now, consistent with the idea that there's reduced survival when we in cells treated with the ATR inhibitor, we next looked at apoptotic signaling. So apoptosis is one of the ways cells can die, a very common way cells die by after DNA damage. So what we did is to look at a cleavage of a protein known as PARP. And so this is a downstream target of the apoptotic caspase enzymes. And this can be looked at using an antibody against PARP. We can detect both the full-length protein, which is, I believe, 110 kilodaltons, and then a cleave form that migrates a little faster, that's smaller in size. And so we can see with increase in the vehicle-treated cells here, with increasing doses of cisplatin treated for 48 hours, we see that there is a increased amount of the cleaved PARP that is observed in these cells. Now, when we pre-treat cells with the ATR inhibitor, now we actually see more PARP cleavage, and particularly if you look at the higher doses here, like 60 and 90 micromolar, there's essentially no full-length PARP left. And so these cells are indeed consistent with the cell survival assays. They're more sensitive to the DNA damage, and they're more likely to undergo cell death by apoptosis. In figure 4b, we simply quantified the percentage of cleaved PARP at these different doses of cisplatin with DMSO or BA21, just to show that this is reproducible. We also looked at the effect of AZD6738. We see very similar results, that it does sensitize the cells to cisplatin. We see more cleaved PARP. We've also looked at cleaved caspase-3, which is a smaller protein, a little harder to detect, and it's upstream of PARP. And we can see that, again, we see more of that cleaved PARP with both ATR inhibitors in the presence of cisplatin. Now, consistent with the idea that cells are dying, one of the things we could do is actually stain the cells for their DNA content by propydium iodide staining, and use flow cytometry to measure cell cycle distribution. So if we look at cell cycle distribution, this peak here corresponds to a G0 or G1 peak, consistent with the idea that the cells are quiescent. We don't see many cells in S phase or G2. But if cells are undergoing cell death, their DNA gets degraded. And so we could look at the cells, what we refer to as a sub G1 population. So the DNA content is less than what would be found in simply a diploid cell that hasn't undergone replication. And so we can see when we treat cells with cisplatin, and particularly in the presence of the ATR inhibitor, we see more of this sub G1 population of cells. And in D, we've quantified that by, I remember now if this is by, oh, it's up there. Yeah, okay, the percentage of cells in sub G1, just to make sure I remember what we did. Essentially quantifying the percentage of cells that are down here. And we can see that in the ATR inhibitor, we see more of that sub G1 population. So what we're next curious about, again, these assays are very short term, looking at within a couple of days after cisplatin treatment. So we next wanted to look at long term. So what we did was to design experiments that could monitor the ability of quiescent cells to divide once induced with growth factors. So what we did was to take our quiescent cells grown in the normal way, grown in confluence. We wait 2 days in low serum, then pre-treat with ATR inhibitor or not, and then expose to, in this case, we used a lower dose of cisplatin to avoid some of the toxicity that we would see. Then we wait 5 days, allow cells to recover. Again, these drugs are present throughout that time. Then we replate the cells at very low density and look at the ability of single cells to divide and form colonies, which we could then stain and count. And so we wait 10 to 14 days for that. And so if we look at simply this clonogenic survival, we see essentially no difference in cell survival. About 40% of the cells at this low dose of 10 micromolar cisplatin are able to form colonies. And in the presence of the ATR inhibitor, that doesn't seem to affect that too much in these Hackett cells. And that's consistent. If we look at cell survival, for example, at lower doses of cisplatin, there's really little effect of the ATR inhibitor. So that's probably not too surprising. But we wanted to use lower doses of cisplatin in these experiments to be a little, maybe potentially more physiological in terms of what a stem cell, a quiescent stem cell, might be exposed to in vivo. We also looked at mutagenesis, and so in this set of experiments, we did something very similar, but during this 2-week growth period, we incubated cells with the drug known as 6-diaguanine. So this drug normally gets metabolized by cells, and it's toxic to the cells, so they can't grow. But if they have mutations at this gene locus known as HPRT, they're unable to metabolize that 6-guanine, and they become resistant to that drug. And so cells treated with, for example, UV, or in this case, cisplatin, we see an increased number of mutants. Normally, we'd see essentially no mutants if we were to do this assay in the absence of any cisplatin treatment, for example. But if we treat cells with cisplatin, we see in the case of vehicle-treated cells, DMSO, a little bit more than 2 mutants per million cells that we plate. So it's again a very low mutation rate. However, when we treat cells with the ATR inhibitor, now we increase that level of mutagenesis. So we see more mutants, more cells that become resistant to 6-guanine, suggesting that one of the functions of ATR in quiescent cells treated with cisplatin is to prevent mutagenesis. And so in addition to promoting cell survival, it looks like ATR has a function in limiting mutagenesis. So we next wanted to look at what ATR might be doing. One of the major functions or one of the main things cells need to do when they're damaged with DNA, or have DNA damage induced by cisplatin, is to remove those cisplatin adducts from the genome. And they can do that through, and as I mentioned earlier, through nucleotide excision repair. So we wanted to look to see if inhibiting ATR had any effect on the rate of removal of cisplatin adducts from DNA. So we did a DNA immuno dot blot assay. So essentially, we treated cells with either DMSO or the ATR inhibitor and the presence of cisplatin. And then at various time points after treatment, we harvested the cells, purified the genomic DNA, spotted it on a nitrocellulose membrane, and then probed with an antibody that recognizes a cisplatin adduct or total DNA. And so we can see over in figure 6A here that about 24 hours after UV or after cisplatin treatment, we see a peak level of cisplatin adducts, and then at later time points, 2, 3 or 4 days later, it starts to decrease, right? So we see less platinum adducts remaining in the DNA. They're getting cut out by nucleotide excision repair. In the presence of the ATR inhibitor, that rate is very similar. So again, if we quantify this, we don't really see any difference. By 4 days, there might be some difference, and part of this is probably the level of some cell death that might be taking place that might really, to some extent, affect the accuracy of our quantitation. So it looks like this increased mutagenesis and decreased cell survival that's affected by ATR in these quiescent cells is independent of the actual rate of removal of UV photo of cisplatin adducts from DNA. So we next wanted to look at another step of nucleotide excision repair, and that's the gap. So I mentioned earlier that once the damage is removed, it leaves a gap in the DNA. We can actually assay that some of those processes by fractionating cells. So we can essentially break cells open, isolate the histones and the chromatin inside the nucleus, and look at proteins that are bound to that material. And so in figure 7, what we've done is to treat cells just as before with the vehicle or ATR inhibitor, expose cells to increasing doses of cisplatin, and then I believe this is 24 hours later, we harvested the cells and looked at a couple different proteins. One is RPA. So RPA is a single-stranded DNA binding protein. So it's thought to bind those single-stranded DNA gaps that are generated by repair. And we can certainly see an increase, a dose-dependent increase in RPA binding to chromatin. Similarly, we looked at pCNA. So pCNA is a homotrimeric clamp protein that helps keep DNA polymerases on the DNA. It increases the processivity of the polymerase. And that, again, is thought to be important for efficient DNA synthesis. And so when you have a gap in DNA that's generated by repair, that has to be filled in to complete the repair reaction and generate a duplex DNA. So we, again, just like in the case of RPA, we see a nice dose-dependent increase in cisplatin levels on the chromatin. And when we inhibit ATR, neither of these are really significantly affected. It looks very similar. However, one of the things that is different is a modified form of pCNA. So pCNA, as I mentioned, is a clamp, but it's known to be post-translationally modified by ubiquitin. So ubiquitin is a small, about 8 kilodalton protein that can get added onto to other proteins. And one of the things this does, in the case of pCNA, is it serves as a signal to recruit specialized DNA polymerases. So most DNA polymerases may have trouble replicating damaged DNA. However, if there are other family of DNA polymerases known as TLS or translesion synthesis DNA polymerases that are, depending on the polymerase, able to replicate across certain types of bulky DNA adducts in either an error-free or error-prone manner. And so what we found here, very interesting enough, is we certainly see this level, this induced by cisplatin in these quiescent cells. So again, it is being induced. However, when we inhibit ATR, we see more of that. So again, we see more of this in the right 3 lanes compared to the left. So again, it looks like when we inhibit ATR, there's more of this modified PCNA in the cells. And because this signal, this modification of PCNA is thought to recruit TLS polymerases, and because some of those TLS polymerases are mutagenic and they're less, they're more error prone, less error free, this might explain the increased mutagenesis we saw earlier on in this paper. So we think that's an important trigger. And we've seen similar things with UV in a separate paper I've talked about. We see very essentially identical results. When we inhibit ATR, we see more of this modified PCNA, and that correlates with increased mutagenesis. And then we've also probed for histone to make sure we're looking at histones, and we can actually visualize all the histones by Pronto staining. And again, figure B, we simply quantified that. So there is a modest difference in RPA levels at the lower doses of cisplatin. Really no effect between inhibiting ATR and PCNA, but when we inhibit ATR, we see more modified PCNA in these cells. Now, one of the questions I think one of the reviewers had was, well, is this PCNA loading association with chromatin dependent on repair? So we inhibited nucleotide excision pair with spironolactone, and consistent with what we predicted, when we inhibit repair with spironolactone, we reduce the level of modified PCNA that's generated in the cell. So suggesting that this association of PCNA with the chromatin is indeed dependent upon nucleotide excision pair generating those gaps in the cells. So this leads us to again a model for how what we think ATR is doing in quiescent cells treated with cisplatin. Again, one of the major adducts induced by cisplatin are adducts between adjacent guanine residues in DNA. There's other adducts that can be induced as well. A nucleotide excision pair can cut out that damage in the form of a small DNA, and that leaves a gap in the DNA that can become bound by RPA and by PCNA. So RPA binds the single-stranded DNA. PCNA is thought to bind the primer template junction there to allow DNA polymerases to come in and replicate across to restore that gap to a normal duplex DNA. Now, single-stranded DNA bound by RPA is thought to be a major trigger for recruiting ATR via an interaction between ATRIP, a subunit of ATR, and RPA. And that in replicating cells, that's again the model that has been put forward and tested by many different groups. Now we think a similar situation may occur in quiescent cells as well, where we don't have replication, but we can certainly generate single-stranded DNA by nucleotide excision repair. And so normally, when we have active ATR, we may have some level of PCNA modification, but it's not as robust. However, when we inhibit ATR, as was shown in the previous figure, we see certainly PCNA is loaded on the chromatin, but we see as more of this modified, more monoubiquitinated PCNA. This is thought, and this is again a focus of future studies, is to look at the TLS polymerases, but we suggest that there may be increased TLS polymerase recruitment to these gaps. And because depending on the polymerase, some of these, again, as I mentioned earlier, are mutagenic, this might increase the level of mutagenesis that takes place during DNA synthesis to fill in that gap. So we think, in the absence of ATR, when ATR is inactive, we will see there's maybe defects in this process leading to increased cell death, as we showed when we looked at cell survival, and increased mutagenic cell survival. So when we look at, for example, the HPRT assay. So we think, again, ATR, in addition to having very important roles in replicating cells, and even in non-replicating in quiescent cells, it plays an important role in regulating how cells respond to DNA damage, including by this very commonly used anti-cancer drug, cisplatin.